Thank you very much. Now, what I want to do is to present to you the results of a feasibility study for a new project that I introduced last year. And it's basically a process for deriving Tier 2 SSROs beyond the Tier 2 pass-fail approach. And before I do so, I'd like to acknowledge my co-authors, Robin Angel from Stantec, Eric Lamb from the University of Saskatchewan, Barry Zadlick from Zadlick and Associates, and Melissa Whitfield Aslan, who is an NSERC IRDC industrial postdoc at Intrinsic. And so the presentation overview includes a brief background for the basis of this research, an overview of the research program, and I'm not going into any technical details of the statistical analysis, because the person who was supposed to give this talk was Eric Lamb from U of S. He is a plant ecologist with a background in statistics. I am an environmental toxicologist with no experience in structural equation modeling. And unfortunately, or fortunately, Eric's daughter came early, so he could not be here. I'll give you a status update on the different approaches that we investigated, and I will give some detail on the PLS approach, the SEM approach, but I'm not going to do anything on the drama approach. I leave the drama to Barry Zadlick. And then I will do proposed next steps, because we did receive additional funding to take this a little further. So the research providers, I think we're mandated by PTAC to present this slide, include individuals from Stantec, Barry, who is he's a PhD candidate at the University of Waterloo, as well as an independent consultant, and Drs. Eric Lamb and Steve Siciliano at the University of Saskatchewan, and then Melissa. And then our research partners include Elizabeth Hersack. I keep forgetting she got married about 4 years ago. And then Chris Mallock from Husky Energy, and he's our scientific authority for PTAC. And then there's myself, and then the funding agencies that are involved. So the background for this project, Alberta regulations had a provision for application of a Tier 2 pass-fail approach to assess risk associated with remediated soils contaminated with petroleum hydrocarbon residuals that remained above the Tier 1 standards. So this whole project only deals with one exposure pathway, and that is the direct contact exposure pathway. There is also provision in the Alberta regs for deriving a site-specific Tier 2 remedial objective, but to date, there is no process beyond the Tier 2 pass-fail approach for doing this. There is evidence that the risk associated with exposure of ecological receptors to soils contaminated with weathered and aged petroleum hydrocarbons is less, the toxicity of the residuals is less, or the risk associated with the toxicity of the residuals is less than that associated with soil contaminated with fresh product. And there's more and more evidence that this is in fact now becoming absolute fact. And as a result, the risk for ecological receptors associated with exposure to petroleum hydrocarbons in soil is often overestimated. Now, the real impetus for this project came as a result of some work that I was doing with Steve Coleman and Tony Knafla, actually. And what I found was that the ecotoxicity assessments that were being requested by clients or proponents could range from anywhere from $24,000 to $212,000, depending on the number of soils that were being tested and the size of the test battery, including the number of species and the number of tests that were being requested. When the soils passed, and that means when the soil toxicity data or the toxicity data for soil samples met the criteria established by Alberta Environment for a specific land use, site closure was supported by other lines of evidence. The cost savings could be high because closure was obtainable. when the soils failed, site closure is not supported, and further remediation of the soils might be warranted or management measures imposed. But the bottom line is, Steve Coleman paid me a lot of money to do an ecotoxicity assessment, and he did it right. He had all the field replicates, he had replicate lab samples, he had replicate chemistry data. He did a really nice job, and at the end of the day, the soils failed, and he didn't even have a remedial benchmark to clean up to. He had to either decide to remediate further or impose management options. And I looked at all of this data that I had, and I thought, surely to God, there must be a way to come up with a site remedial objective given all this toxicity data. But then we started looking at the toxicity data, and it was really challenging to make heads or tails out of it because, and Tony, you might recall this, that one of the most toxic soils was in fact one of the control soils. Toxic meaning it had the severest effects expressed in all the tox data. There was no pattern to fertility, there was no pattern to the physical chemical characteristics, there was no pattern with the concentrations of hydrocarbon. So I thought, this is just noisy data. There must be information here somewhere. And I started investigating the potential of teasing out the signal from the noise. That's how the statisticians like to express it. And I was working with a woman who was doing a postdoc at University of Toronto on a metabolomics study. And she had generated a huge amount of data that was really noisy. And so she was investigating these different statistical methodologies for reducing the noise in the data and highlighting or emphasizing the signals. So I got talking to her, and she was interested. She was just finishing up, and she was really interested in taking this further. And at the same time, I started getting visions at night that there might be a way out of here. And so I thought, and I started talking with Barry, who was at the time looking, as part of his PhD thesis, looking at ways of deriving environmental quality criteria. So I thought this was going to be possible, that there could be a process for deriving site-specific remedial objectives at Tier 2 when a Tier 2 pass-fail approach failed. So the first thing I did then, the challenges before me were to get individuals to share the vision. And that was a bigger challenge than I thought. But I did manage to find 3 statisticians who were working in their own lines of research on similar issues, and they were keen and interested in seeing whether or not the tools that they were developing could be applied to this project. One of the stumbling blocks or challenges we had was that this is a very data-hungry process, and there's no formal way of accessing all of the toxicological data that are out there that are related to petroleum hydrocarbon fractions, because nobody's collecting it in one place and making it readily available. However, I did have a large amount of data in-house, so I didn't view that as too much of a stumbling block, but in an ideal world, I can see that as going forward to have this database acceptable for users and in the areas of contaminated land management would be very beneficial. And in fact, I had some discussions early on with representatives from PERD who were keen on actually taking this on at the time. So we proceeded with looking at developing models, because this is the vision I had. If we could develop predictive models that would, in fact, you could go to a site, given the physical chemical characteristics and the contaminant profiles in those soils, you could predict the biological responses for those soils. And then you could use the distribution of those predicted effects at these sites to either focus your remedial efforts where it was needed, exclude soils that didn't require further remediation because they in fact met the predicted level of risk associated that was acceptable. And to be fair, I actually tried this at a site and it worked beautifully. And we were in fact able to exclude 150,000 cubic meters of soil from further remediation, which the ecotoxicity assessment cost him about $167,000. The cost of remediating those soils that were contaminated would have been about $2 to $3 million. So it was hugely successful in this one instance that I actually looked at. But that was a site-specific initiative. We were asking the question, what if we develop these models that could be applied to any site? And that's what we want, that's ultimately where we want to go. So what I want to do now is to present the results of the feasibility of, or the investigation of the feasibility of 4 approaches that we took. And the first one was simply to look at the distribution of the geometric means of the no observable adverse effect concentrations and the lowest observable adverse effect concentrations that resulted from an ecotoxicity Tier 2 assessment for the pass-fail. Okay, we knew the soils failed, so let's look at the NOx and the low-X and see if we can use the geometric mean and the distribution to actually come up with a site-specific remedial objective. And then I provided subsamples or a subset of the data that I have to both Barry Zadlik and to Eric Lamb and to Melissa, and each one of those individuals took a different approach to investigating the potential of developing predictive models. And Barry, the acronym for his investigation was data reduction and model averaging, and he's going to speak to that in the next presentation, so I'm not going to present any details on that. Eric Lamb looked at structural equation modeling, which is a process used in plant ecology. Again, where large amounts of data, noisy data, can be looked at quantitatively. And then Melissa Aslin was looking at partial least square multivariate regression analysis. So the rationale for the geometric mean approach then was simply to take the data from the Tier 2 ecotoxicity assessment and could it be used to derive an SSRO by examining the distribution of the ranks of the geometric means of the NOx and the low-X. You can do it, and what we did, we took the data for an ecotoxicity assessment with 4 plant species, 2 invertebrate species. We had 8 fine-textured site soil samples with 3 reference control soils. So in the end, we had in excess of 25 toxicological endpoints from which to make an assessment. We looked at the distribution of the geometric mean, and that's what is shown in this slide. And what we found was the derived number of 1153 for the 25th percentile, which is the policy decision for residential and agricultural lands. The derived guideline was below the Tier 1 standard. And then when we looked at the same 50th percentile for the industrial and commercial land use classes, the number again was below the Tier 1 standard. So there was no relief for using this approach, and we still think that this approach, there's an inherently large amount of conservatism in taking this approach. So we actually didn't pursue that much further than that. For the partial least squares regression approach, the rationale for using this approach is that multivariate analysis can be used to describe the relationship between a multivariate predictor matrix. So you could have a matrix with pedological characteristics and contaminant variables, and a response matrix, which is the Y values. And this approach allows you to assess the strength of the relationship between X and Y, and it can be used to predict future unknown Y values based on known X values. So if you had done a site survey and collected a lot of soil samples to have them chemically characterized, which is normally what they do for phase 1, phase 2s, and for risk assessments. And then, but you didn't do toxicology testing on those soils. Could you build these models to predict what those effects would be based on, which is the X matrix, based to predict the Y values, which is the biological responses, using this methodology? This is the type of information that you can get. This looks at the PLS models were derived using leave one class out cross validation procedures. And the model fit was evaluated, and the predictive ability of this model has yet to be evaluated. But using these procedures, Melissa did look at a preliminary data set and showed that the actual predicted number of progeny produced when plotted against the actual number of progeny, the model wasn't really great at describing or predicting those effects. But she then went on and looked at all of the other endpoints and demonstrated the importance of non-contaminant variables in explaining the variability in the biological response data. And this is these are the model parameters for earthworm progeny production. And when you look at what figures fairly prominently in describing the relationships between the X matrix and the Y values, what's clear from this presentation, this is just one endpoint, but it was consistent for most of the endpoints that we looked at, is the relative importance of the non-contaminant variabilities in describing these, the variability in the responses. So for this example, you can see that total carbon, total nitrogen, and soil moisture, salt, magnesium, and sodium adsorption ratio were as important or more important than any of the contaminant variables. So to conclude then for this approach, this is our conclusions to date. It is possible to link multivariate soil properties to some of the ecotoxicity endpoints. The predictive power of these models is likely to be inadequate for soils when the soil properties differ significantly from the soils from which the data that are used to derive the models differ substantially. And the approach shows great promise on a site-specific basis, but we're not convinced that the utility for cross-site evaluation. The good news, and this was sort of surprising to me, maybe not to Melissa, but the results of her findings were corroborated by the findings of Barry Zadlick's analysis using DRAMA. Now for structural equation modeling, it was investigated also as a potential process for driving SSROs, and there's two components to this approach. One is the measurement. It includes the measurement model with indicator or latent variables, and the other component is the structural model, and that's the paths between these variables. And just to give you some idea of what he's talking about, or we're talking about, for the measurement model, what he did was he took an approach whereby he looked at all of the individual endpoints that were measured, and he came up with a way of modeling a predicted aggregate species response. So this was useful for data reduction. And then he used that aggregate species response to turn around and estimate an IC25 or an IC50. And this is the first attempt at doing this based on the model that you saw previously. Now in the report, I think in the end there were something like 24 models were investigated, different combinations of things. And so I'm just giving you a glimpse of the types of things he could do, or he did. And then he looked at the predicted IC50, which is the dashed line in this slide, and the IC25 that was predicted for the aggregate species response for F3 in soil. Now the structural component then is the path linking these different the measurement variables with the structural variables, which include the pedological characteristics as well as the contaminant variables. And so he looked at soil quality, including total carbon, total nitrogen, or organic matter content, because these were the critical variables that were most influential. Texture was incredibly important, as was the concentration of fraction 3. It's not always fraction 3 that's critical. Sometimes it's none of the fractions dominate, and it could be total petroleum hydrocarbons. So one of the advantages of this approach is that he could use a confirmatory factor analysis to aggregate the multiple endpoints into a single latent variable that can then be incorporated into standard nonlinear regression models to estimate any ICP value. And this had the advantage of reconciling divergent endpoint data, which is usually a challenge. You either rely on best professional judgment, although Barry has another way of looking at this challenge, and you have to make some assessment on what endpoints are giving you useful data and which endpoints are not. The SEM also showed promise in terms of cross-site modeling, because he was able to explain the aggregate species response with an R-squared value of 0.7, so which was sort of surprising when you use those models that were developed for one site or one set of data to predict what was happening in an independent data set. And the only problem was that the models that he used failed all tests for model adequacy. And that was, he thinks, is a function of just the fact that we didn't give him enough data to look at for this approach. And so he wants to pursue this analysis on a larger data set. So in summary, the Tier 2 pass-fail approach is of limited value when the site soils fail. I don't know if you're that familiar with it, but if you've ever been the recipient of a report where your soils all fail, you just want to cry. But what we've discovered from this analysis is that failure to meet the Tier 2 criteria is not necessarily a failure due to toxicity when a substantial portion of the observed toxicity is attributable to non-contaminant variables. And those variables could be soil texture, organic matter content, sulfur, and pH. And Miles, interesting enough, of that partial least squares analysis, elemental sulfur is the one variable that shows up in every single tox test as an important explanatory variable. We're pretty sure that statistical approaches that were used quantified the relative importance of the PHC contaminants versus the non-contaminant variables with respect to, I don't like to say toxicity, I think in this case we're dealing with effects, because it's hard to believe that organic matter content could be perceived as behaving like a toxicant. And the probability of making a correct decision is greatly increased when you apply the appropriate statistical tools for interpreting the toxicological information from these ecotoxicity assessments. And that's, I think, when you don't get the answer you want, you don't use statistics to get the answer you want, you use statistics to better understand what your data are telling you. So the next steps for this fiscal year from the AUPRF funding is to apply PLS and DRAMA and the SEM analysis to a larger data set. There were challenges with the data set that we provided to these statisticians, and Barry will go into that in some detail. And we want to finalize by the end of this year a predictive model for each of these approaches. And then we want to apply the models to site data that's independent of the model building data set and predict a distribution of effects on that site. And then the ultimate verification and validation will be to go out and collect soil samples from that site and test the accuracy of the predictions. And to do that, I will need a site that is contaminated, that is exceptionally well characterized in terms of having the right physical chemical characteristics measured and a sufficient quantity of measurements. I've got one site in Manitoba that are volunteering. I have some reservations about that site, but if there are any in Alberta, I would be pleased to hear from you. And then, based on the data from this verification and validation, I suspect the model refinement and recommendations might be warranted.